All right, and so in this last section of our basic hematopoiesis, so our generation of the different immune cells from the bone marrow, what we see is that neutrophils are one of the most common cell types in the immune system that are the nucleated cells or the white blood cells that would be inside the bloodstream. But these cells are also one of the most short-lived. So this animation is actually showing how different cell-bound and soluble factors stimulate our hematopoietic stem cells to actually become a neutrophil. So we'll take a look at this. So first you have release of TPO, and the TPO is a stimulating factor that's called thrombopoietin. And this can be expressed by bone marrow stromal cells, and it can interact with the hematopoietic stem cell to start differentiation of that hematopoietic stem cell into the megakaryocytes. So megakaryocytes can be utilized to produce things like thrombocytes. So this factor here, the thrombopoietin, can be utilized to also produce thrombocytes or platelets within the bloodstream. So remember, our red blood cells, our erythrocytes, and the platelets are going to be the most common items that are going to be found in the bloodstream, especially the erythrocytes that carry the oxygen and carbon dioxide. The platelets are the very small cells, and they're actually kind of cell fragments. They're enucleated as well, so they do not contain a nucleus, and they contain most of the blood clotting factors. So they're utilized to recognize damage to the endothelial layers that actually are lining the blood vessels, and they can cause the clotting cascade to occur so that they can keep the blood vessels from rupturing and causing massive bleeding within an area. So at this point, you have the common myeloid progenitor cell that's going to arise from the hematopoietic stem cell, and this is going to give rise to a granulocyte or a macrophage progenitor, and this is done through this GM-CSF molecule, which is the granulocyte macrophage colony stimulating factor. So these are small cytokines or growth factors again that are going to interact with some of these myeloid precursor cells or progenitor cells and cause them to start differentiating into the granulocyte macrophage progenitor. These guys are going to reproduce, and then they are going to become the neutrophil progenitor as well. And then other small factors like the granulocyte colony stimulating factor are going to interact with the neutrophil progenitors to produce the mature neutrophil that gets transported into the bloodstream. Neutrophils are cells within the immune system that are one of the first responders to the inflammatory response. And so if there's an infection with like a bacterial infection within the patient, or if you have damage to an area, so some physical trauma, or even some forms of cancer, will start recruiting the immune system to that area, and the neutrophils are one of the first responders. So we saw a YouTube clip already in the class where a neutrophil was actually chasing a bacteria, and so the bacteria actually produce these small chemokines or small chemical signaling molecules that are attracting the neutrophil to it, so it can actually engulf the bacteria in the process of phagocytosis and basically eat the bacteria or chew them up and make them go away. So this is another YouTube clip. It's a neutrophil that's moving to the site of infection, and so we'll just go ahead and play this. But this is just to show that the neutrophil, once it's trying to go to the site of an infection, it's got little receptors that are displayed on here that will recognize certain factors that get expressed within the bloodstream. So here you can see that the neutrophil is actually interacting with the endothelial cells, and they actually will roll or crawl along the surface, and they're looking for damaged regions. So you can see that they're moving much slower through the blood system. You can see the small erythrocytes and the flow of the blood actually moving very, very quickly. So platelets and erythrocytes move very quickly. Bloodstream's over here in this case. This would be an area that has some damage, maybe an infection, or if you sprain your ankle or something like that and there's some tearing or damage to the area. you will recruit neutrophils to the area to deal with that infection. So as the neutrophil is going along, it's got its receptors, and it will actually stick and start to roll on the endothelial layer. So the endothelial cells that line the capillaries, you can see that in damaged areas, they're going to produce signaling molecules that actually will attract and they will lock the neutrophil in place. So it gets trapped to an area that is going to require the sensory information. And this sends signals then to the neutrophil that cause it to release specific cytokines of its own. And so it does this, so it's releasing small molecules, and when it does this, it's actually releasing a small protein called the heparin binding protein. And this causes the endothelial cells to leave enough room that that neutrophil can slip into the surrounding tissue, where it can start investigating and moving around looking for damaged areas, looking for possible bacterial infections of the tissue, and start dealing with these invaders and causing phagocytosis. So the next topic that we want to talk about is innate immunity. So we're surrounded by a myriad of microorganisms which live around us, on us, and inside our body cavities. So we've already been exposed to this in our bacterial section and in our viral section, where we've seen like the host species of bacteria can actually affect whether we're prone to disease states or not. So our microorganisms are going to include bacteria, viruses. They also include fungi, which we haven't talked much about yet, and then protozoa as well. And we use the Plasmodium falciparum, or the malarial parasite, as one of these examples for disease states that can be caused by protozoa. So also some worms are parasitic as well and can cause infections. So all these different sources of material can cause disease states within humans. And so our first line of defense against invading creatures from the outside that can cause disease states is something called our innate immunity. And so let's take a look in a little more detail at the immune system. So it's divided into these two major phases, our innate defense system and our adaptive defense system. So innate immunity is going to act immediately or very shortly after an induction period to a foreign substance or a foreign entity that is going to invade our system, like bacteria, virus, protozoa. And it's going to recognize specific patterns on indispensable molecules of groups of microorganisms, and these are using pattern recognition receptors. So these are specialized receptors that are on the innate immune system cells, which are going to be predominantly the neutrophil that we saw in our last diagram. This is one of our innate defense mechanisms. So these guys are set up to recognize foreign invaders like bacteria, be able to chase them down, recognize these specific patterns that show on specific groups of bacteria or viruses or the parasites, and be able to engulf them by phagocytosis. So other natural innate immune cells are going to be things like macrophage and natural killer cells that are going to come to the defense, to the initial defense, or be around kind of sitting there like little generals waiting for these disease-causing organisms to invade our system. The innate immune system is less effective than adaptive immunity, but it helps to keep our pathogen level down while we can mount an adaptive immune response. So the adaptive immune response really involves the production of antibodies that are going to be very, very specific to a specific attack by a microorganism or by exposure to certain compounds as well. So it requires about 5 to 7 days of an attack by a specific microorganism before it becomes fully active. So if a person is already immune to an attack, this response time is going to be shortened to 3 to 4 days. So what the adaptive immune system is going to do, it's going to recognize unique structural features of the pathogen. So it actually is becoming very pathogen specific at this point, so that it can mount an immune response to a specific pathogen. And it does this through antibodies or on T cell receptors. So our production of the lymphoid system that's going to end up producing both the T cells and the B cells is going to be critically important for our adaptive immune response, whereas the myeloid cell lines that produce the macrophage and the neutrophils and the natural killer cells is going to be more prominently used for innate defense. In comparison to our innate defense, our adaptive defense is going to be very highly specific and effective. It's going to completely eliminate the pathogen from the host. Whoop, this should be pathogen. It shouldn't be a pathogen. And for this system to actually work, you have to have cooperation between both the innate defense system and the adaptive defense system. So they're not working totally independent of each other. They actually have coordinated effort to achieve a maximal immune response. So we're going to spend a little bit of time talking a little bit about the innate defense mechanisms, and then we will get to the adaptive defense mechanisms and see how our body actually produces antibodies. So here are the key cell types that are responsible for innate immunity and their functions. So we've talked about epithelial layers. These are our primary barriers within the body. So they're going to form the major linings of all the body cavities. So you can have multi-layered epithelium like we see with the skin, and you can have single-layer epithelium like we see with the intestinal lining. So basically our epithelial cells are providing a barrier. They will also produce things like antimicrobial peptides. One of the most common is called alpha defensin, and we'll take a look at the structure of that protein here in a few minutes. They also tend to secrete mucus, so not so much for our skin cells. Our skin's pretty dry, but if you looked at the inside lining of the intestine, or the inside lining of the lungs, or the inside lining of the stomach, you would see that there's a mucosa that actually gets produced, and you have secretion of the mucus. The mucus actually is going to be the area, it provides a protective insulative cushion for the lining cells, the epithelial cells that are going to be lining the intestine or the other body cavities, to keep water layers from just kind of running through here and washing away and causing damage to the cell layer. So it's providing kind of a cushion area of slower moving things. It's also a way to help trap certain compounds close to the epithelial cells. So it tends to trap these antimicrobial peptides, for example, within the mucus layer so they don't just get washed away. And this can help if you have like binding of a pathogen, for example, from food molecules that you may have eaten. This is going to help those antimicrobial peptides actually work and mediate some response to a bacterial pathogen or a viral pathogen that might be trying to enter the system. There's also production of other things that will help maintain production. So fatty acids typically are going to be emitted on the skin surface, and these fatty acids are actually very important components. So free fatty acids actually act like detergents, and so it's almost like we have this detergent layer that can help break apart bacterial membranes. We're protected by it because we excrete it into the dead layers that make up the surface of the skin. And so the fatty acid release at the surface of our skin doesn't damage our skin tissue because we have a very thick dead cell layer that's at the surface. However, it will damage bacteria that might be sitting at the surface of our skin cells and cause them to die before they can actually invade into the skin tissue, especially if you have some skin damage. So like if you get scratched or something like that, you still have these fatty acids that are secreted in the skin to help mediate a protective response. So they actually have kind of an antiseptic property instead of having an actual antimicrobial property where they're attacking the microbes in a specific way. Other areas will have specific regions where they alter specific physical characteristics that are also going to prevent the survival of many types of microbes. So the stomach, for example, has very, very low pH, which will cause the denaturation and the rupture of many microbes, including many of the protozoa, fungi, and bacteria will get broken down under these extreme conditions of pH. There's also a host of digestive enzymes that are secreted both into the stomach and the small intestine. These are also further breaking down the building blocks that are required to maintain the structure of specific organisms like bacteria, fungi, and viruses. You also have fluid flow that's going to sweep materials across an area so that they don't get stuck and have a lot of time to enter into the epithelial layer. So for example, in the digestive tract, there's constant flow that's moving the majority of the components through the system very quickly, so that you get absorption of nutrients from the diet as you have these digestive enzymes working, but any bacteria or larger molecules that would be in here are going to get shunted or moved past this area pretty quickly. Many of these cell layers also have sweeping cilia associated with them. These are areas that have high surface area, and so these cilia act as areas that can trap materials, so they can trap nutrients for entry into the cell, but they also can act as a sweeping motion to help larger molecules just move past. So these sweeping cilia actually can keep things like the airways and the lungs very clear from larger pathogens like bacteria from actually sticking to any one area, and so they'll move them out of the system. This is also true within the intestinal system as well. So directly underlying the epithelial layers, you're going to have connective tissue. So this is going to include a lot of fibroblasts and extracellular matrix that keeps everything connected. So it's going to join the epithelial layer with the underlying muscle tissue that's going to be needed for the actual system integration. Within this connective tissue area, you also have the presence of many of our innate immune system cells as well. So you're going to have a population that includes macrophages or monocytes. You're going to also have neutrophils present, and you're also going to have natural killer cells. So all of these guys are involved in the phagocytosis of small organisms like viruses, like bacteria, like protozoal parasites, and even viruses as well. And so they'll recognize specific surface markers on these different pathogens, and they will undergo or mediate this process of phagocytosis. And once they do this and they start breaking down the bacteria or the other pathogen, they will also start secreting some of these inflammatory mediators. So these would be small molecules or small proteins like cytokines or chemokines that we've been talking about that help stimulate the growth of new immune cells from the hematopoietic stem cells in the bone marrow. So you get that going on. But it also will start recruiting more of our inflammatory response mediators to the site of the infection. So these chemokines and these cytokines act as basically these big beacons that say, hey, we have damage in this area. We need to recruit more immune cells to the area. And so we saw our example video in YouTube about our neutrophils, and this was actually the neutrophils responding to these types of signals within the tissue, being recruited to an area that requires a little bit more help. Maybe it's been damaged some way, causing inflammation, like if you sprain your ankle and cause tissue damage to an area, or there's been infection due to bacteria entering the area, or possibly even the formation of cancer cells, are going to start recruiting immune cells to the area to try to take care of the damage that's being produced. So both our macrophages, monocytes, and neutrophils will also emit reactive oxygen and nitrogen species that help damage the pathogen and start digesting them. Our neutrophils are also producers of antimicrobial peptides like the alpha defensins, and so we'll talk a little bit more about this. And then our natural killer cells will lyse or break down virus-infected cells as well. So these guys can actually recognize infection. They start recognizing the proponents that are going to make up the viral coat protein, and these get expressed on the surface of the infected cell. And so they stand out a little bit like a beacon for our natural killer cells to start recognizing, so that they can start killing damaged cells before they can actually produce the viral load. And so these are very important parts of our innate immune response. So this is a cross-section of the intestinal cell, and we'll use this as an example for how the innate immune system really gets set up. So here you see our layer of epithelial cells lining the intestine. These are ciliated epithelium, and you can see this is the luminal space. So this is where the food molecules would come down during our digestive process. This would be the underlying connective tissue area. So you can't really see the extracellular matrix in here or the fibroblast cells that line in here. But you have access to the bloodstream, so blood capillaries would be pretty close here so that you could take digested food products right into the bloodstream. And you also see a bunch of macrophage cells that are kind of underlying this area. And these guys set themselves up in kind of this orderly array so that they have quick detection of any invaders or pathogens that might be getting into the underlying tissue area that might cause an infection. The other thing you see here along our epithelial cell layer is that some of these epithelial cells are very specialized cells. So you can see right here, these cells are actually the mucin secreting cells. So they're going to help produce the mucus layer that's going to form on the surface of the epithelial layers that help protect the epithelium from invaders. They also kind of act as a sticky area that can help trap very small molecules and nutrients that are going to need to be recruited and passed through the epithelial cells to get nutrients from our diet into the bloodstream. However, you want to keep larger organisms and pathogens from actually coming in and going through this system as well. And that does occur in part due to the mucus layer, where you actually have accumulation of antibiotic peptides like the alpha defensins. So let's take a closer look here at the intestinal lumen. So you have production of the mucin layer by these very specialized cells in the epithelium of the intestinal lining. So you can see this kind of holds some of our bacteria at bay. They're too large to kind of fit in this mucin layer, but you still can recruit and obtain and trap small nutrient molecules like proteins and carbohydrates that start breaking down in the intestinal area, and you can pass the monomer building blocks through the epithelial cells on into the bloodstream behind here. So if we also take a closer look at how the epithelia is working during a damage event, so if you see, like say you eat like a chicken bone by accident, and the chicken bone is pretty sharp and it's going through your intestinal cell, and say it actually pokes through the intestinal barrier. So now the underlying tissue layer is going to be exposed to bacteria that are in the luminal area or other possible pathogens that were in the luminal area of the intestine. So they break through this layer, and now here's a damage event. So that's really what our macrophage are actually for. They recognize specific surface markers from these groups of bacteria. This will cause signaling into the macrophage to start producing these chemokines or cytokines that are going to help recruit and cause an inflammatory response to the system. So this will help recruit other innate responders to the area, such as our neutrophils or our natural killer cells or other macrophage or monocytes. And they will come in and start trying to mount an immune response, so you get some inflammation going in this area as you recruit more and more cells to the area. And they will start producing antimicrobial peptides, such as our alpha defensins. And they'll also start undergoing this process of phagocytosis, where on the inside, in these huge vacuoles or lysozyme areas, they will start to produce reactive oxygen species or other reactive nitrogen species that will start breaking down and damaging the pathogen as it's ingested into these innate immune cells. So this is just a picture of the peptide that makes up the alpha defensin. It's a very, very small peptide. It's only between 2 and 6 kilodaltons in size, and so an average protein in comparison would probably be upwards of 50 to 300 kilodaltons in size. So these are very, very small proteins, and they're mediating an antibiotic effect or defense mechanism for the actual cells. And so I think we'll stop here for this section and then continue on talking a little bit more about the activity of the defensins, and then we'll start getting into our adaptive immune response.